Working in the field of myeloma, one can see the progress that we have made over the last 20 years, the concerted effort of many across the board. And as I mentioned previously, I do think there's a fraction of myeloma patients that are curable, and we just need to get smart about how we're going to expand that fraction. Ideally, we would have a future where there's a therapy that lasts 5 days, just like we have for hairy cell leukemia, and then all patients, can be cured. But we're not there yet. But we can extend the lives substantially. And I think given some of the results, for instance, from the MAIA study, there's a lot of patients who are not transplant candidates, elderly, that will have a close to normal life expectancy once they receive some of this best therapies. I'd like to see a future where stem cell transplant goes away. Despite my affiliation and my affection for it right now as an effective tool, I'd like that to go away. I'd like to see a future where we don't deal with long-term toxicities, a future with peripheral neuropathy absent from the lives of myeloma patients. Patients are going to live many years and have to deal now with the effects of that peripheral neuropathy. And a future where we can use dexamethasone equivalents without the toxicity of dexamethasone. We're getting closer. It's going to take time, unfortunately, but we're getting closer. And I think the ability to treat myeloma will only continue to improve. Maybe we'll include CAR T cells and frontline therapy for patients after a quick induction, and would allow us to just get patients back to their normal lives instead of spending so much time in our clinics.
Progress in the past has been palpable. Patients are living, or progression-free survival is 3 to 4 times longer than it used to be, and many patients have myeloma as a chronic illness. But there is much work to be done. If you look at age-matched controls who don't have myeloma, those patients live much longer than patients who have myeloma, at least in the data that exists in the SEER database. Suffice it to say, we have more work to do, and the 2 areas of most promise are targeted therapies, the therapies that are based on targeting the abnormality in the genomic profile that's present in a given patient with multiple myeloma. And I'll mention a clinical trial called MyDrug, which is about to begin, where patients are profiled for their genetic abnormality. And there are multiple arms in this clinical trial and novel agents that are going to be tested that are directed at those abnormalities. So for example, if patients had 11-14 translocation, they would have venetoclax. If they have FGFR3 abnormality, there's a specific drug for that target, etc. In this particular trial, it's in relapsed myeloma. And so how can you do a novel agent like this in relapsed myeloma when we have so many accepted regimens? What is simply being done is patients are profiled, they get a standard acceptable level of treatment for their relapsed myeloma, in this case ixazomib and pomalidomide, and then they get the addition of the novel agent that's directed at their particular abnormality. And we examine whether the abnormal clone of cells is reduced when you add in the targeted therapy. This is called MyDrug, and the point is it will give us leads for what might be targeted therapies we could use in myeloma, and then we'll have to do tests later to prove that. So targeted therapies on the one hand, and venetoclax in the 11-14 translocation myeloma patients is already the first targeted therapy that likely will come to get FDA approved. The other, besides targeted therapy category, is the immune therapies. And I'll just mention many immune therapies, but new monoclonal antibodies such as elotuzumab, new immunotoxins where you have antibodies bound to toxins, so BCMA, B-cell maturation as a target, antigen bound to an immunotoxin such as an auristatin. That medicine is already in clinical trials and showing promising results. Perhaps the other 2 immune therapies I'll just quickly mention are the bispecific T-cell engagers, or BiTEs. We know that in leukemia and lymphoma, the BiTEs are active and even now, in some cases, FDA approved. But in myeloma, we have the BCMA antibody binding to myeloma cells and an antibody attracting the CD3 positive T cells to the microenvironment of the tumor, hoping in so doing to get more activity and less side effects. So monoclonal antibodies, or immunotoxins, and the BiTEs or bispecific antibodies. And the last is the CAR T cells. Many studies now, much excitement about CAR T cells, where you harvest the patient's T cells, infect them with a vector which has them target, in most cases, BCMA, activates their T cell receptor, and in fact allows for their expansion ex vivo into large numbers and subsequent transfusion back to patients, as I like to call it, as their own immune army. And this concept of CAR T cells is really been very exciting in myeloma. The most exciting news is the depth of response, even in patients who have had 6, 7, 8, 9, 10 prior lines of therapy. We can get MRD negativity, fewer than 1 myeloma in a million normal cells. We have work left to do, however, because we need to make this procedure safer, and we need to make it also so that the benefit is more sustained. We're at the earliest level of the CAR T cells, so there's much excitement, and there should be, but I think we also need to recognize that there's much work to be done to improve the efficacy on the one hand, the safety on the other.
The future for multiple myeloma, I think, is even more exciting. We have lots of interesting and exciting approaches. I think what we've seen in the last year or so is a really good and relevant target in multiple myeloma, and that being BCMA, so the B-cell maturation antigen. We've seen remarkable responses with the conjugated antibody. We've seen incredible responses with the BiTE, which is off the shelf, and we are seeing incredible results with cellular therapy now for the first time in myeloma. So CAR T cells in multiple myeloma, we've seen really nice results with durability of responses in patients who've achieved MRD negative disease state. So these are all very exciting, and I think we will have to figure out, obviously, when things like this work very late in disease, all of these are going to be brought earlier on in disease. And at least with the BB2121 product, we're already bringing it up front, not quite up front, but up front in the high-risk patient population or at the time of first relapse, and in relapse myeloma after 1 to 3 lines of treatment as well. Those are ongoing trials which are actually asking the question on durability of responses with a cellular product such as BB2121. There are a whole bunch of other interesting targets as well. And I think, to me, what's most interesting in the small molecule inhibitor space specifically is MCL1 inhibition. You saw a lot of nice data with venetoclax, which is a BCL2 inhibitor. And the BCL2 inhibitor in combination with bortezomib or with carfilzomib has shown remarkable results. Now, most of us know that in multiple myeloma, MCL1 is actually the predominant proapoptotic pathway. And if you can block that pathway, you're going to see incredible results with an MCL1 inhibitor as well. So we have early data in phase 1 trials with an MCL1 inhibitor, and we'll have to see, as long as it's not a very toxic drug, that's a drug which I would be looking forward to seeing the data on. Other drugs which are being studied and which may get approval for the treatment of myeloma are drugs with a very different mechanism of action, like selinexor. It's a nuclear transport export inhibitor, and this one, in combination with dexamethasone, has in fact shown nice responses in a penta-refractory. Now we're talking about penta-refractory patients. So penta-refractory includes patients who are refractory to pomalidomide, lenalidomide, bortezomib, carfilzomib, and daratumumab. So if you can have an oral drug which can give you a durable response rate, that would be incredible as well. We'll have to see what the data with selinexor looks like, and whether the combination of selinexor with either bortezomib or carfilzomib is here to stay. But these are all interesting compounds which are going to continue to change or keep moving our goalpost for myeloma. And hopefully we will see a very significant subset of patients wherein we're able to control this disease for a long time, which in effect would be curing this disease.